All right. Hello, everyone. I have 11 a.m. Central Time on my clock, so we'll go ahead and get started. Welcome to the Algal Bloom Action Team's webinar series focusing on harmful algal blooms. I'm excited to have two great presenters here today, and we're going to be talking about cyanobacteria in the context of climate change. For those of you who are unfamiliar with the Algal Bloom Action Team, we are a collaboration of water professionals, researchers, and educators from the 12 states in the North Central region. And we are made up of individuals from Cooperative Extension, from the North Central Region Water Network, and from the Water Resources Research Institutes at each of our 12 states. And I have a list of our partners there, including the North Central Region Water Network and NIWAR, the National Institutes of Water Resources. We do these webinars on a bi-monthly basis. So we feature different HAB researchers from across the region and beyond every other month, featuring their research. So today we're going to be featuring two researchers who are going to be talking about cyanoHABs in the context of climate change. And we do have a next webinar coming up in August. They are always the first Wednesday of the month at 11 a.m. Central Time. We also put out a suite of resources, and you can find those on our website at northcentralwater.org/HABs. Part of our resources, in addition to this webinar series, are a suite of fact sheets, including a fact sheet that we recently debuted for pet owners and livestock owners on harmful algal blooms and what you need to know. That can be used with your outreach with various stakeholders. We also host a annual harmful algal bloom research symposium. That's a virtual 2-day symposium that's held in January each year. So this coming January 2025, will be our 5th year of that research symposium. So look out for that. So we will get right into our researchers today. We have 2 presentations for you, each one about 20 minutes in length, and then we will have dedicated Q&A afterwards. This session is being recorded and will be posted to our website at that URL you can find there, northcentralwater.org/HABs. We will draw the questions from the Q&A from the Q&A panel. So you should see that at the bottom of your screen. So make sure to submit your questions for our researchers, our presenters there, and we will go through them at the after both of the presentations, excuse me. We do also have the upvote feature enabled. So you can upvote other questions that others might have that are similar to yours. We will try to get to those questions that are the most popular first. If you are having any audio issues, you can click on the audio or the microphone and go to switch to using your phone. If you're also having any other issues, you can put them in the chat and we'll try to work those out or troubleshoot with you. All right, let's get to our first researcher, which is Rebecca North. Rebecca is an assistant professor at the University of Missouri and got her education at the University of Waterloo in Ontario. Rebecca's lab has a lot of research in terms of focusing on the stressors of nutrient cycle, bioavailability, and primary production on cyanobacteria and on water bodies overall. She has a lot of research that goes into the implications of varying light and nutrients and temperature on cyanobacteria. So I'm excited for her to share some of that research with us today. I'll stop sharing and hand it over to you, Rebecca.
Thank you. All right, everyone can see my slides okay?
Looks great.
Great. Yeah, so I'm an associate professor at the University of Missouri, but I'm speaking to you today from Berlin because I'm currently on sabbatical and I'm serving as an IGB Senior Research Fellow in Germany, and I'll present some experimental work that I've done here as well. So as mentioned, I did my PhD in Ontario in Canada, and the definition of limnology I was taught, a little tongue-in-cheek here, was the study of lakes located within 60 miles of a major university campus, but not during the winter. And so, yeah, while this is a bit of a joke, it also is serious in the fact that most of what we know about lakes is data that's collected during the summer. And so this is based on, yeah, undergraduate student availability, but we do tend to have this kind of summer-centric lens and focus for our understanding of lake ecology, and then, of course, more recently, cyanobacteria. And a lot of this was based on these kind of traditional paradigms. So this is showing chlorophyll A concentrations, which is a proxy for phytoplankton biomass, showing the kind of bimodal peak we'd expect out of dimictic lakes or, northern lakes that have ice cover. And so we kind of plan our monitoring or sampling season to encompass these two peaks. So one occurring in the spring and the second one in the fall. And as we all are probably familiar, we expect to see our cyanohabs kind of peaking in that late summer period as well. But we know that our seasonality is changing, and unfortunately, our winters are getting shorter. So this unmonitored season is literally changing beneath our feet, and we don't have a lot of baseline data to understand what's going to happen related to these changes. So unfortunately, we are on a trajectory to be able to lose most ice cover by the end of the current century. And so when we start thinking about what's going to impact our phytoplankton communities, and then specifically the cyanobacteria, which are a portion of the phytoplankton, we typically look at these kind of four major controls. So as primary producers, they need light to be able to photosynthesize. So light is important. We also know that temperature, water temperature, is an important control. And then Silvia will talk more about this in her talk, but we'll also relate to nutrients. So looking at nitrogen and phosphorus as two macronutrients influencing phytoplankton growth. And so these three things are what we call kind of bottom-up processes. And so I don't think any of us are going to talk about it, but we are aware that there is top-down or grazing processes that controls phytoplankton populations as well. So today, for this presentation, I'm going to focus on the two physical controls, changes in light and temperature. And so we were very fortunate to be funded by NSF to start a new project that actually just started sampling in May this year called Thin Ice. And so this is looking at warming winters and changing lakes. And so in this project, we're using a space for time gradient to look at impacts of climate change on lake ecology, and then specifically looking at cyanohabs and the production of cyanotoxins. So we have a gradient of lakes that range in latitude or elevation. And so, from a pessimistic perspective, as we lose ice cover, we could be going from these nice dimictic lakes that are covered in ice and snow to a future of lakes that are no longer covered. So in fact, one of the lakes I work on here, Lake Stecklin, used to have ice cover like 5 years ago, and now consistently doesn't get ice cover at all. So there's already some lakes that are on this trajectory. And so we know when we do have ice and snow in the winter, that we get this inverse stratification. And so all the phytoplankton tend to kind of live up here close to the light. And this actually, helps them, the convective mixing or the stratification helps keep them suspended up there where they can get light. But what we're starting to see more so is this delayed and intermittent ice cover. And so kind of, these stops and starts and reset buttons to what we'd expect out of a continuous ice cover is now happening intermittently. And then when you get to the warm monomictic, and so I do most of my research in the state of Missouri, and so that's pretty much doesn't get a lot of ice cover, so they're warm monomictic lakes. So with no ice cover, you can actually get deeper mixing. And so you find that if it's a really, really deep lake, you can get your phytoplankton are being dragged down deeper into deeper depths in the water column. So losing ice and snow does have impacts on temperature, but it also has significant impacts on light. And so we can kind of hopefully see this here, where in these dimictic lakes, how much light the phytoplankton see depends typically on the depth of snow and ice, but most of the time they're kept up here because of the light in this convective mixed layer. Whereas when we lose the ice, then they can get dragged down into the dark waters. So this is showing the light here, and they would spend more time in a more monomictic situation in darker waters than they would have if they'd had ice and snow. So that's just a little thing that's kind of hard logically sometimes to wrap your head around. And so, of course, as we see changes in temperature, we're going to see changes in the mixing or the stratification of what we expect out of lakes on a seasonal basis. And so these are some conceptual diagrams showing what we predict to happen. And so this is, the full year on the x-axis from January to December. What we currently have in the northern states of dimictic lakes are shown in gray, and then maybe the future, or what we see in Missouri as warm monomictic lakes, are shown in black. So in the future, you would see in warm monomictic, we have warmer water temperatures. We would have, as I explained, changes in these mixing patterns, which is going to affect the mean light, or the amount of light that phytoplankton experience over a 24-hour period. And so again, this could decrease as they're getting dragged down into these lower, deeper depths. And then, of course, we expect to see changes in nutrients related to changes in stratification. It's also going to affect the phytoplankton populations. And so here we're showing kind of the three major groups. And so, as we talked about, they kind of traditionally had this kind of bimodal peak to what we expect out of phytoplankton, with most of the cyanobacteria showing in the cyan color here, happening in the late summer months when the water temperatures are warmest and stratification is the strongest and most stable. But in more monomictic conditions, we tend to see kind of more consistent dominance of cyanobacteria on a year-round basis. And so starting to look to the future, are we going to see more of our dimictic lakes experiencing those types of things? And what's that going to mean for cyanobacteria or for cyanohabs? So in my presentation today, I'll suggest that life in a lot of ways is getting better for cyanobacteria. So they're definitely not going away. And we might need to kind of start thinking outside of the box. We might be looking at the wrong times, the wrong places, and for the wrong types of cyanotoxins. And so I'll provide some data and papers that we've produced on cold water blooms. So that's the wrong times. Ubiquitous cyanotoxins, so looking beyond just microcystin, which is the most common, to include also looking at toxins, cylindrospermopsin, anatoxin, and saxitoxin, and what drives those, and when one is high, the others can tend to be low. And then also some experimental work that I've done here in Germany, looking at microcystin persistence. So kind of a disassociation of the actual toxin itself from the phytoplankton that produce it. And so all this kind of, weight of evidence might indicate that what we kind of think of as being, green equals toxic may no longer be true. And so we need to start being aware of that and think about how to mitigate it. So the objectives of the talk today are the impact of changing light and water temperatures and on cyanohabs, and kind of thinking ahead to what will happen when our ice cover disappears. And so one of the seminal papers that we all probably are aware of about cyanohabs is Blooms Like It Hot. And so again, we expect to see them, kind of late in the summer at those warmest water temperatures. But last year, a group of international colleagues published this paper in LNO Letters. And so this is through GLEON, the Global Lake Ecological Observatory Network, where we started kind of talking. We have a winter limnology working group and started having some conversations about how we also see blooms in cold conditions. So put together some global data to show that that is the case. And so most of the rest of the data I'll show you is from Missouri in the US. So if you're not familiar, I call it the middle of the middle of the middle, or the flyover zone. You might more recently know it from Netflix, produced the show Ozarks. So Lake of the Ozarks is one of our study lakes. And I will keep using the term lakes, but really I'm talking about reservoirs. So most of these water bodies have a dam on them, and they're actually reservoirs and man-made. What's really nice about Missouri is that it has a gradient and trophic status. And so this means that in the north, it's 70% agricultural land use, and the lakes tend to be greener, or what we refer to as being eutrophic. Whereas the south, you can see here, there's a national forest in the south, so it's about 60% forested, and we can get some oligotrophic or really clear blue lakes in that area. Though if you're looking for them, it's also not very hard to find cyanobacteria. And so commonly our messaging to the public is, kind of look before you swim. And if it looks nasty and green, then it could potentially be toxic. But yeah, I'm going to suggest that maybe we need to kind of expand our view on this. So this is some work by Yusuf Olay. He just defended his master's thesis, and he looked at 4 different types of cyanotoxins over a variety of reservoirs in the state of Missouri. So here we're showing oligotrophic reservoirs, so really low nutrients, low chlorophyll as the clear, all the way up to hypereutrophic as the really green ones with lots of nutrients. And so you can see here for the toxin microcystin, we see kind of what we'd expect, that the highest concentrations occurred in the hypereutrophic reservoirs. And the letters over the bars indicate statistical significance. So if the letters are different, then they're statistically different. So in this case, the highest, significantly higher microcystin concentrations in hypereutrophic reservoirs. However, there was some surprising results. And so the next one here, B, is showing cylindrospermopsin concentrations. And so although these concentrations tend to be pretty low, you can see that the highest concentrations are occurring in fact, the oligotrophic reservoirs. So what we thought of as the nice, clear blue reservoirs. Anatoxin seems to occur kind of continuously. It's ubiquitous, and it doesn't seem to differ that much, based on nutrients and trophic status. And then saxitoxins can occur in our most eutrophic and hypereutrophic reservoirs, but tend to be rather low. So kind of looking at different toxins and expecting different relationships. We can also start thinking about different times when we can expect to see blooms in cyanotoxins. So the shoulder season, the spring and fall, and the winter season obviously are understudied, as I mentioned, lack of undergraduate availability. But we do know that these blooms in some cases can produce toxins. And although most of us aren't polar dippers and aren't in the water and swimming this time of the year in the shoulder seasons in the winter, we do drink our water year round. And we can also, get experience or get exposed to cyanotoxins through spray aerosols. So boating or just being near or on the water. So it's still important to understand what's happening on a year-round basis for the management of our lake and reservoirs. So this is some data from 2 urban reservoirs in Columbia, Missouri, done by Emily Kinzinger as part of her master's thesis. You can see here she's looking at microcystin toxins and cylindrospermopsin toxins and differentiating them by temperature groups. And so, cold tends to be 0 to 15 degrees Celsius, and warm are these warmer summer temperatures. And so, most of us, when we first started doing this, we expected to see the highest concentrations happening in August and September when we had the highest water temperatures. But instead, you can see the peak concentrations are happening in these kind of shoulder seasons. So for microcystin, it's happening here in the late fall, and cylindrospermopsin, it's happening in like January and February. And so when we looked at these concentrations, we started to look around at the phytoplankton populations that were present at the time and tried to figure out who the toxin producers were. And so for this data here in November with these high microcystin concentrations, there was actually very little cyanobacteria present in the water column at the time. So only 4% were cyanobacteria. And then it was dominated by a type of cyanobacteria that's not a known toxin producer. Same story with cylindrospermopsin. So these really high concentrations that we see happening in February and March is when there wasn't even any cyanobacteria in the water column at the time. So there seems to be this lag effect or this disconnect between when we see high toxin concentrations and when we find the phytoplankton, the cyanohabs that produce them. And so as we were kind of puzzling over this and trying to figure out what was going on, there was this review paper published in L&O Letters that talked about the microcystin cycle. And so this is their conceptual diagram here showing all the places that microcystin can come from or be stored in and all of the different fluxes. And that got us kind of thinking about cyanotoxin degradation and persistence. And so maybe there is a discoupling between the actual production of the cyanotoxins. Maybe in some situations, they're hanging around in the water column longer than we think. And so at the end of this paper, they suggested that future investigations should focus on sediment fluxes, drivers of biodegradation, and the seasonal dynamics. So this was a great launching pad to do some experimental work. And so this is how I spent my sabbatical. We did some experimental incubations. And so this is again at a Lake Lab facility on Lake Stecklin with my grad student, Fatima Larib. We took sediment cores. So here is showing a sediment core, 3/4 of it was full of sediment from the bottom of Lake Stecklin. And the first bit was water. We treated these like if anyone's done internal phosphorus loading experiments, it was very similar. So we had references where we just took it right from Stecklin. We spiked some with microcystin RR just to get some high concentrations. We spiked some with Planktothrix, which is the type of cyanobacteria that has like this red pigment that tends to bloom in the deep waters actually of Lake Stecklin. And then we have some with no sediment at all, so only water. And so we set these all up and then we hung them, these 4 different treatments in the top of the water column. So high light and high temperature, middle of the water column, medium light and medium temperature, and then the bottom of the water column. So really low light and low temperature. and then went and measured, and so drew subsamples out of this, the top of the water on a regular basis, and ran it for 20 days. And so here's some experimental results. You can see here we started on day 1 and measured microcystin RR concentrations in micrograms per liter on a regular basis throughout the incubations. I'm showing the different treatments here, but the story is even in the ones that we just spiked with the microcystin RR, where you can see that at this high light and high temperature, so the top of the water column, the microcystin degraded pretty fast, right? So just sitting there, it declined pretty quickly at a rate of minus 0.2 micrograms per liter per day. The incubations that we hung at the bottom of the water column, so at low light and low temperature, you can see much less of a steepness of slope here. So they didn't decline as rapidly. The rate was only about 0.5 micrograms per liter per day. And you'll note that the ones that we started off kind of at high concentrations that we spiked, even by the end of 20 days, they were still around 4 micrograms per liter. And so I think this probably differs by state, but at least in Missouri, it's a general guidance is 2 weeks. So they say if the water looks really nasty and you expect cyanohapis occurring, wait to swim or wait to fish or wait to do any activity for 2 weeks. So yeah, just kind of showing that if there's low light and low temperature, like there is in the winter, that they can stick around for significantly longer than that. So hopefully at this point, I've managed to convince you that, in our future, life might be getting better for cyanobacteria. And we can kind of maybe think about this space for time, and that Midwest reservoirs may be a precursor of what to expect in a climatically different future. And to kind of, expand our lens or what we would expect to find cyanotoxins. We might be looking at the wrong times, wrong places, and for the wrong cyanotoxins. So there's a variety of exposure ability in the off seasons as well. And so rethink, kind of whether or not green does equal toxic, and whether or not we should be looking at other indicators. And so with that, I'd like to acknowledge all the projects and the funding sources that have contributed to this. And I guess at the end of the webinar, I will welcome any questions that you have.
Wonderful. Thank you, Rebecca. I appreciate it. I know I've long heard the idiom in climate change, there will be winners and losers. So I think that's an interesting thing, in that perhaps cyanobacteria are sitting over there as winners. We're going to transition over to Silvia Newell today. She's going to be talking a little bit about nitrogen as a driver for cyanobacteria growth. Silvia is the director of Michigan Sea Grant, and she also serves as a professor at the University of Michigan School for Environment and Sustainability. So I won't read her full bio, but it's up there for you, and I will hand it right over to her.
Okay, thank you so much. I hope you can still hear me okay.
Yes.
Fantastic. And we're all good with full screen sharing?
Looks great.
Well, thank you so much for having me here today to talk about my work, not as Michigan Sea Grant Director, but as a researcher working on harmful algal blooms. I only moved to Michigan about a year ago, coming from Ohio, and so a lot of my focus is still on Lake Erie and some of the lakes in Ohio, but is becoming a global problem everywhere, especially inland lakes in Michigan, and so my focus is expanding a little bit. So Rebecca gave a really nice introduction into HABs. I just wanted to emphasize that in addition to cyanos liking it hot or maybe cold, that it's really increased nutrient loading that is causing a lot of the harmful algal blooms that we're seeing, at least cyanobacterial ones. And that to date, regulations on trying to mitigate harmful algal blooms, particularly in the Great Lakes, and through the Annex 4 agreement, the Great Lakes Water Quality Agreement, that those have targeted reductions in phosphorus, but haven't really addressed any other kinds of nutrient reductions, particularly nitrogen. And this is sort of a question of timing of when this was developed and signed versus what data was available at what point. And this dual nutrient control strategy document had not yet come out from the US EPA that was showing scientific support for both nitrogen and phosphorus criteria. And so why do we care about this? Why dual nutrient management? Well, as both phosphate and inorganic nitrogen concentrations increase, we get really large blooms that are highly toxic. And the idea was that if you controlled phosphorus, that would make the bloom much smaller, and it may make a very small bloom, in fact, but it could be a very small but highly toxic bloom. And to me, that's not a super great alternative. If you just reduce nitrogen and not phosphorus, you might end up with a possibly non-toxic but large nitrogen fixing bloom, larger nitrogen fixing bloom. That doesn't seem like a great idea anyway. So my stance has always been that dual nutrient management makes the most sense so that we end up with small and non-toxic blooms or lower toxicity blooms. And the idea that nitrogen might compensate for, or nitrogen fixation might compensate for reductions in external nitrogen loading is certainly true, but nitrogen fixing is very expensive and it requires breaking a triple bond. And that's becoming less and less relevant across water bodies in the temperate US at least, because we have such increasing reactive nitrogen in the world. So kinds of dissolved inorganic nitrogen, including ammonia, urea, nitrate, have really taken off since the Haber-Bosch process allowed us to make bombs and then fertilizer. And you can see from this chart over here on the right from Erisman and Larson that the total amount of nitrogen, reactive nitrogen in the world has just increased exponentially. We're at about 4 times the natural level of biological nitrogen fixation at this point. So nitrogen in fertilizer is just simply a lot more bioavailable than nitrogen gas in the air, and our shift has been toward an even more usable form or usable forms of nitrogen. So I don't know about globally, but at least in the US in 1996, after the building or the bombing of the federal building in Oklahoma City, it became a lot more difficult to buy ammonium nitrate, and we see this shift towards urea and anhydrous ammonia, but not just in the US, across the world. And urea and ammonia are both super usable by cyanobacteria. There's a nitrogen control gene cascade about what gets used first, and ammonia is by far the easiest kind of nitrogen to use, followed by urea. And then a distant third is nitrate, and then way down at the bottom of the ladder after everything else is exhausted is nitrogen fixation. And this is interesting when you think about it in the context of the kinds of bacteria that are around forming these harmful algal blooms now. Lake Erie had harmful algal blooms in the 1960s and 70s, but they were nitrogen fixing. You can see in this picture over here on the left that Annabena, right, has these they were heterocysts, now they're heterocytes. Same thing with aphanasamenon. These are special nitrogen fixing cells that prevent gas movement across the cell membrane and allow it to block out oxygen and fix nitrogen. And these are really, really, really expensive to make. It takes about 16 to 18 ATP per nitrogen fixed. And that's just not really great if you have an alternative. And the blooms that we have now in the western basin of Lake Erie are actually non-nitrogen fixing. They lost the genes for it. They're no longer capable. It's dominated by microcystis, which is not capable of nitrogen fixation. There are later blooms in Lake Erie, especially in the central basin, of things like Dolichospermum that can fix nitrogen. But when we talk about the big green habs in the western basin of Lake Erie, this is what we're talking about. And so why do we care at all about, dual nutrient control? It's this toxicity, right? And what happens is that the toxins, if you're looking at the structure of them here, if you look at the microcystin up in the left-hand corner, you can see there's a lot of nitrogen. There are 10 nitrogens per molecule of microcystin, and there are 0 phosphorus. And so because there's no phosphorus directly involved in the toxins, it does play a big role in controlling the amount of biomass that's there, but a cell that's super stressed about any nutrient, might have cascading impacts, but in general, it's the amount of nitrogen that controls the amount of toxicity. And so the Great Lakes Habs Collaborative from the Great Lakes Commission put together this document on harmful algal blooms and nitrogen, identifying the major knowledge gaps that we have. And these are some of them, and I will address them today. Thinking about what are the impacts of changing external loads on blooms and toxicity. What's the importance of internal nitrogen cycling, both in the water column and in the sediment? What is, in terms of internal nutrient cycling in the water column, what's, happening with nitrogen turnover? And could that be the missing piece for modeling toxin concentrations? And I'm going to argue that I think it is certainly a way to do it. And then there's a need for nitrogen reductions now that we have better and more data on nitrogen. So this is showing the western basin of Lake Erie. I think this is the 2017 bloom. It was a very large bloom that year. It's got a summer bloom dominated by Microcystis. It's largely because you can see the Detroit River coming out of the top here that's pretty much blue, right? And that's about 95% of the water. And then about 5% of the water comes from this stream over here on the left that's just showing bright green in Maumee Bay, because the Maumee River is only 5% of the water, but it's about 50% of the nutrient load. And this correlation between phosphorus coming in from the Maumee River and then the size of the bloom has been the basis of this decades-long emphasis on peat loading. But it turns out that nitrogen is also important, and it's not just reactive phosphorus that's correlated with the bloom. There's also a correlation between the kinds of nitrogen that's there. So it's this proportion of non-nitrate nitrogen that has been increasing in the Maumee River over the past few decades, and there's a correlation between that and the size of the bloom as well as how toxic the bloom is. And so my group has done a series of studies over the last few years, and I'll just give you the highlights of a couple that address those knowledge gaps. And in this one, we're going to talk about internal loading in terms of sediment nutrient release and processing in the western basin of Lake Erie. And we had 4 sites that we were looking at coming out of the mouth of the Maumee River into Maumee Bay here. That's this MB18 and sort of working its way westward. The northernmost site here, WE4, is actually most impacted by the Detroit River. But when I showed them on a graph, it'll be starting with Maumee Bay and ending with WE13 over here, sort of in the middle part of the western basin. And my graduate student, Ashlyn Bodecker, sampled every site in spring, early summer, late summer, and fall of 2016 and 2017. And this was an Ohio Sea Grant funded project to my collaborator, Mark McCarthy. So we were looking at sediment core incubations in western Lake Erie. And the way that this is shown here, because this is N2 gas production, so it's sort of backwards of how you want to think about it. If there's a positive bar here, it's actually showing that it's a sink for nutrients, because it's showing production of N2 gas through denitrification, which is the reduction of nitrate all the way to N2 gas. That's important because it takes the nitrogen away from being this, usable reactive form. So you can see that most of the year, most of the time, in both a low bloom year of 2016 and a high bloom year of 2017, the sediments did serve as a sink overall for N2 gas, for nutrients. So as denitrification was happening, and denitrification was higher in a lower bloom year, which is maybe not what you would expect, but the residence time of the western basin is rather small. And when there are fewer cyanobacteria, they tend to be more diatoms, and they tend to sink and stay in the western basin and provide more organic carbon for denitrification. Whereas the cyanoblooms are very light and they get pushed out into the central basin, and there's just less organic carbon around for the denitrifiers to actually use to denitrify. And then when we look at the balance of all forms of nitrogen except N2, looking at all the dissolved nitrogen forms here, so ammonium, nitrite, nitrate, and urea. Now these graphs are backwards because these are dissolved inorganic forms rather than a gas. And so if the bar is negative, it's overall, a sink. And if it's positive and stacked, you see that that part is a source. You see the sediments are pretty consistently a source of ammonium at most of the sites most of the time, but overall the denitrification is enough to balance it out and be a net sink for nutrients, although maybe not as strongly as we would like. And then moving from the sediment to the water column, as we're thinking about internal nitrogen cycling, we measure ammonium uptake and regeneration. So as you have cells that are being broken down and remineralized, that release of ammonium we are calling regeneration, but it's not just ammonium, it's also phosphate and carbon and everything else as a cell is broken down and broken up. And then we have the uptake of ammonium because it's very easy to add ammonium and measure the uptake of it. And so we focused this study in a different subwatershed of the western basin of Lake Erie into Sandusky Bay, which is shown right here on the map. And the Sandusky River watershed is also mostly agricultural, and it receives very high nutrient loadings. It's just that the overall discharge of this river is much smaller than the Maumee River in terms of its contribution to Lake Erie proper. But what you see here in terms of the timing of the HABs, which are different actually, they're Planktothrix in Sandusky Bay, or they were at this time, is very similar to what Rebecca was showing, where our peak biomass over here in summer or even, in late summer, is not at the same time as when our toxins are. And we see toxins here in earlier in spring and in the beginning of summer. And what you see in terms of the nitrate availability is that this is lining up with when the nutrients are around. So toxins get made when nitrogen's high. It's not when biomass is peaking. When biomass is peaking in the worst part of a bloom, cells are starved for carbon, nitrogen, and phosphorus. They're turning on carbon acquisition genes, phosphorus acquisition genes, nitrogen acquisition genes. They are sort of desperate for food at that point, and that kind of stress does not lend itself to making microcystin or toxins in general at that moment in time. Which is kind of ironic because it sounds like the purpose of toxins may be to protect cells from super high biomass concentrations that end up making hydrogen peroxide. That may be the purpose of toxins. So, all right, so then we look at not nitrogen concentration, because at this point in the middle of the bloom, right, if you look at the bottom, the nitrogen concentrations are very low. So internal nutrient cycling must be really important then, was our hypothesis, to supporting these blooms. And so we went out and we measured how fast does the nitrogen turnover, how fast is ammonium getting regenerated or remineralized at these various stations that are going from the mouth of the Sandusky River over here on the left, all the way to going out into Lake Erie on the right at the Bell Station. And this is showing over time in a summer, going from early June into late August. You can see that what the ammonium that's actually there is higher than what we measure, between 0.2 and 1.2 micromoles, and that's how much is coming out per hour. So when we measure the concentration, it might be almost zero. We might not even really be able to measure it, but it's being turned over and released so quickly in the late summer that this is the true measure of bioavailability of nitrogen. There's just a lot of cells competing for it at that point. And so this is a sort of busy table here, but what you see is that at the very sampling dates that we went out, this is looking at the Sandusky River loading and how much nitrogen is coming in. And if you just look at the TN column here, you can see that there's still quite a bit for this week of June 5th. There's 11 metric tons of nitrogen coming in. That's dropped pretty strongly by the end of July down to 1.5 for the week. And by the mid to end of August, it's like 0.2 to, it's basically around 0.2 metric tons a week. It's very low external loads coming in at that point when the biomass is peaking. And so we see then the comparison with how much nitrogen is being regenerated at that time over that week. We've scaled it up to metric tons here. See in the far right column, how much regenerated nitrogen or ammonium we're getting. And in June, it's only about 2 to 5 times the external load. By the end of July, it's 25 to 50. And by the end of August, it's about 1,000 times higher than what's coming in external loads. So this turnover of internal nutrients is really what's helping sustain the bloom during peak biomass. And so we thought, okay, if this is what's representing true nitrogen availability, then maybe we can use HABs, or maybe we can use nitrogen turnover to start modeling HAB toxins. And we have some data for Lake Erie, but it's not quite enough to make a good model. These are pretty intensive, incubations and measurements to make. And so we thought we'd start with a smaller lake here in Grand Lake St. Mary's, Ohio. We picked Grand Lake St. Mary's because it was at least a decade ago in the 90th percentile in the US for TN and TP concentrations of loading coming into in the streams coming into the lake. And it was the second most toxic drinking water source in the United States. It's pretty much got a year-round Planktothrix bloom, and most of the time it has pretty high microcystin levels. And so we decided to go ahead and try to make a model of habtoxins over the course of 3 years in Grand Lake St. Mary's through 2021, 2022, or sorry, 2020, 2021, and 2022. And this turned out to be a really apt time to do it because the weather was so different in those 3 years. We had a very high bloom in 2020. You can see, if you look at the middle panel here, might be the easiest. You can see the measurements of microcystin are pretty high. 2021 had a really hard freeze and very little rain, so we ended up with very low microcystin toxins, and then they increased back again in 2022. And so we had a good range of concentrations to be able to look at with the model. And you can see that this model that my collaborator Jason Dahl put together shows the predicted microcystin in the little circles and then the measured microcystin concentration in the triangles. And you can see that they are pretty close together. And this study was involved monthly sampling at all of these sites that my collaborator, Stephen Jackman, who's a professor at Wright State University, the Lake campus on Grand Lake, actually had students go out and measure things pretty frequently every week. And then we did the incubations once a month. And so you can see over here in the left, the inflow of a major tributary to the lake, which is pretty representative, that there was just a lot of water, a lot of rain coming in 2020, sort of a middle amount in 2022, and then this low amount in 2021. And again, that's reflected over here in the, you can see the total microcystins panel showing pretty high concentrations in 2020, low in 2021, and high in 2022, or medium in 2022. What's really interesting, though, is that in 2021, when it finally did start raining in June, if you look at that top panel of chlorophyll, you can see that the numbers jacked up again. So the biomass did come back when it started raining, when the nutrients came back, but for some reason, the toxins never did. And that's not because they didn't have genes. It was actually, you know, toxic strains were there if you look at the bottom panel showing the MCY, but they just never started making toxin. Presumably, our hypothesis was because they just didn't have the available nitrogen. And if you look at the nitrogen regeneration, down here is the third panel down, you can see that the numbers were higher in 2020 and 2022, but they just never really got high in 2021, along with that, with the rates. So we are hopeful that this might be a useful tool going forward to try to predict microcystins. But like I said, the incubations are somewhat time and labor consuming. So we're limited by human power on that. And we're hoping to find enough predictive variables to try to look at nitrogen regeneration that we could then nest or incorporate into models for broader microcystin productivity. What's really exciting about this, and I know I need to wrap up here, but that year that it didn't rain very much in 2021, if you look over here on the right in terms of algal biodiversity and the phytoplankton community, you can see that in 2022, there were some diatoms that kind of like it eutrophic and not very much diversity at all, even in the spring bloom. But in 2021, when it didn't rain very much and there were those reduced loads, we already saw increased biodiversity. You see dinoflagellates and yellow-green algae, golden algae, and green algae, along with the diatoms. So there's just a lot more biodiversity over here when the nutrient loads are reduced, and that's an immediate effect in the same year. So it's making us very hopeful that as we're able to reduce nutrient loads to the lake, that we will see some more immediate effects and not be, you know, completely delayed by legacy nutrients. So thank you so much. I really like to acknowledge all of my amazing co-authors. There are several students in here, including Joe Davidson and Justina Hampel and Ashlyn Bodecker, who were the students who led these projects. My co-collaborators, Mark McCarthy, Stephen Jackman, Jason Dahl, and George Buller-John. So I want to thank all of them and thank both the Ohio Sea Grant and the Water Resources Center of Ohio for funding these projects. And I'm happy to take questions, and my email is up here.
Wonderful. Thank you so much, Sylvia. I really appreciate it. We do have quite a bit of questions that have come through, which is great. So I'll look at those. As a reminder, I do see some questions coming into the chat. Just make sure to enter those questions in the Q&A and upvote those questions that you see other folks ask, and we can get to those as soon as we can. I believe this first question is from Justin and is for Rebecca. At what depths in the water column would you expect to see it? Question came in. Would you expect to see a decrease in the cyanotoxin concentration?
Yeah, well, the factors that break down the cyanotoxins are like UV light. So it'll depend, it's not, the same depth for every lake. It'll depend how transparent or clear your lake is. If it's really clear, it'll be deeper. If it's not so clear, it'll be more shallow. Also microbial activity. And so bacteria like warm temperatures. And so again, it will depend if you're like stratified and where that threshold is. So yeah, not an absolute depth, but it will be dependent on temperature and light, basically.
Sure. This question is for Sylvia. Toxins are made when nitrogen is high, not when biomass peaks. Does that mean toxin thread is high before you see the HAB? To a layperson, what does biomass peaks mean?
So I think it was pretty cool actually that Rebecca showed the same thing, that toxins were highest not during peak biomass, but before and after. Peak biomass just means when there's, the most cells per volume in the water. And it was something like microcystis, that's really easy to see because in a really bad bloom, well, they always float on the surface, but it can look like spilled paint on the surface of the water. And I've seen it get so thick in China that you can just reach over and like scoop it up with your hand and hold it. So that would be like true peak biomass. And even though there aren't as many toxins per liter necessarily or per cell, you still, with that many cells, you might still have enough toxicity that you'd want to, not drink it, could still be dangerous and harmful. But yes, it does mean that toxicity might be a problem before you really see that being that bad. But I just want to emphasize that there are other kinds of cyanobacteria like Planktothrix or Limnothrix that live further down in the water column. They don't form that spilled paint, and people don't get as worried about them even during peak biomass because they picture it like the spilled paint, and then if it doesn't look like that, they think it's not really a problem. And so I just emphasize the need to start testing and potentially warning people in late May, early June even, and make sure that we're protecting the people who are going to beaches.
Yeah, absolutely. And so this kind of question goes to that note. And Rebecca, you had a note about this too. So how would y'all as researchers think about suggesting the messaging so that it's not always that, green or the spilled paint, as you talked about, Sylvia, equals toxic, as we y'all have noted that it doesn't always correlate in terms of when the toxicity is high is when you look at it and go, oh, yuck, I can see the paint on top. How might you suggest from a messaging standpoint, we address that as folks working within water?
Yeah, if you want, I can say my guess, but I think at this point, yeah, it's, we'll see. I think measuring the toxin concentrations themselves might be the best approach as opposed to using proxies for it, because as we said, there's a lot of assumptions that are made that might not necessarily translate. So unfortunately, there's no quick and easy way to measure toxins, which will probably be the next question. So, there is like these, Abraxas and other companies make like these dipsticks and things like, there is things that you can find out results within 20 minutes. They don't give you, really low concentrations and stuff, but at least as a first measure to see whether or not. But then, of course, as we found, there's also the different types of toxins. So yeah. All times, all places, all toxins? I don't know, Silvia, how would you respond?
I would say a couple of things. One is that it is important in places where that toxicity has been high or has been increasing and monitoring is possible and available to either start sampling or encourage the watershed group or lake association group to start sending in toxins or sending in water samples for testing earlier, again, like late May, early June. But I would also, in messaging, show pictures of water, say, in a bottle, as opposed to showing just like painted water on the surface. Because if there's a bad Planktothrix bloom, if you pick up the bottle and you show it, it looks green, right? It's just that it doesn't look as green from the surface of the water, especially with sunlight reflecting off of it, etc. So if you pull up that bottle of water and it looks green, like, that's when it's time to really worry about it.
That's a great suggestion. I haven't heard that one before, Sylvia, though I have heard on this, noting just the, in terms of messaging, be aware this body of water, this reservoir, this lake has tested high in the past. Be sure to check ahead of time. So it's almost like highlighting the history, since you can't always tell by looking, which I think is an interesting point as well. Another question. Oh, go ahead.
Sorry, many states do have like a beach guard website or something similar where people can go and check, and just having a sign that says this is tested, high in the past with a QR code to that website, I think would be really helpful messaging a lot of beaches.
Yeah, I love that idea. Absolutely. Especially as folks are getting more and more comfortable with those QR codes, looking those up, and more folks are having, smartphones. Ryan has a question for you, Sylvia. Can you reiterate your theory on why toxins are produced in microsystems?
So it's not my theory, but other people have shown, and especially led by Greg Dick's group here at University of Michigan at the Cooperative Institute for Great Lakes Research. It's a cooperation between the University of Michigan and the NOAA Great Lakes Environmental Research Lab, have shown that there may be a connection between toxin production and protection from like superoxide radicals, hydrogen peroxide, basically dangerous oxygen forms that when you start having so much photosynthesis, right, in a very small confined, well, not confined, but a small area, right, a small volume, you get a lot of oxygen production. When you have that much oxygen production, you can really form these dangerous radicals that are hard for the cell. Exactly how toxicity prevents that or protects from that, I'm not entirely sure. I'm not sure we really know the mechanism yet, but it does seem to be that cells that are toxic strains are better protected from that.
Yeah, thank you for going over that. I appreciate it. Greg is curious, do N and N to P ratios play any role in toxin production?
Right. So when you have high extra nitrogen around,
Yeah,
that's when you see more toxin production.
thank you. I believe this question actually came in for Rebecca. Any consideration of herbicide concentrations in lakes repressing photosynthesis or herbicide resistance cyanobacteria compared to other phytoplankton as an additional variable, especially in ag-dominated area of Missouri?
Yeah, so actually the same working group that wrote that cold water blooms paper also did one on organic nutrients. And so in that, we also included all herbicides and pesticides. And we found that there was a lot of nitrogen and phosphorus in a lot of these products. So, you're adding it to try to kill the phytoplankton, but in essence, you're actually doing like a nutrient addition experiment and adding a lot of nitrogen and phosphorus as well. So I think that would be one of the primary things is the nutrients.
Interesting. Thanks for that. Santina is curious, how does a dual nutrient approach translate in terms of the management actions needed to reduce nutrient inputs? We have been assuming that agricultural practices to reduce P will also reduce N, and have been seeing N inputs increasing. I think this would probably be just a little bit towards both of you, though I know it is more specifically to you, Sylvia.
Yeah, so it depends on the agricultural practice, obviously, but many of the things that we're doing to treat P will also treat N, right? Anytime that we are holding back water, we're also holding back nitrogen, any kind of edge of field, buffer, strips, wetlands are at least at some point taking up and removing both nitrogen and phosphorus just through different pathways. Obviously all those things can eventually become saturated and, need maintenance, etc., and could become a source. but that's true for both nitrogen and phosphorus. Were there things though that involve like some of the edge of field strips with like iron in them, like those kind of treatments, that's only going to address phosphorus.
Gotcha, yeah. But when you think of something like controlled drainage, if it's holding water back, it's gonna hold water back with N and P is kind of your point there. This question is for Rebecca. Does climate change encourage extracellular toxins relative to intracellular toxins?
So extracellular toxins are usually produced when the cyanobacteria die. So, if they self-crowded, like if they end up forming these really thick, dense blooms and then they cut each other off basically from light, then they could be dying. So it could happen, as we get more cyanobacteria dominating the water column, you could theorize that you would get more dying, and therefore more production of extracellular toxins. But basically, yeah, they come hand in hand. I guess one other comment on the earlier one too about adding herbicides. So it's pretty common practice in Missouri for drinking water treatment plant operators to put copper sulfate or other things in the water when it starts turning green to try to kill the phytoplankton. But what that actually does is it bursts open the cell. So now, in addition to the extracellular toxins you have in the water, you now have opened the cells and added the intracellular toxins. So, you increased your toxin concentrations in the water. So a lot of things that kill the cyanobacteria actually will increase the toxin concentrations.
Interesting, thank you. All right, I think we have time maybe for one, maybe two more questions, and I'm just looking at some questions that are maybe we haven't addressed before. Val has an interesting one. Are you concerned about the lack of monitoring for guanine toxin, formerly anatoxin A, sorry, I didn't know the rename, and related risks to dogs and other monogastrics, including humans?
I mean, obviously, it's a thing that we worry about coming online in the future. At the moment, I mean, Rebecca showed more data on different kinds of toxins, so she might be a good person to address it, but we aren't seeing as much anatoxin yet, in my understanding.
I guess my question on Val's is just, are you indeed noticing that there's less testing for anatoxin A and some of these other toxins that we know are there, and while you're normally seeing the testing for microcystis?
Yeah, I think, every state will have different monitoring and testing programs, right? And so they'll be doing different toxins in different ways. So I think as more of these things become commercially available through these Abraxas kits and ELISA kits and things, then you'll see more testing. But if it's so specialized and expensive, then I think, yeah, it would be less done on a monitoring basis. So it is my understanding, but I'm not in that world, that each state handles like a complaint of a harmful algal bloom differently and will test for different toxins and have a different response rate, etc. So there's not even a universal mechanism for that, even for microcystin.
Thank you. I think we have time maybe for one more question here. I believe this question is for you, Rebecca. Going back, what are your thoughts on why Silendospermopsin levels were so high under cooler and less nutrient-rich conditions? Were the Silendospermopsin, and I might be mispronouncing it, sorry, I've heard you say it, producers simply being outcompeted by MC producers in warmer and more eutrophic waters, or do you think the toxin-degrading bacteria were less active? Do you have a kind of a theory there for why you might have found that?
Yeah, this is what's really complicated about the cyanobacteria and the cyanotoxins is the same cyanobacteria can produce both microcystin and Silendospermopsin. So they're not necessarily separated, or you can't link them one-on-one with species. And so, yeah, that could happen. But also we know that Silendospermopsin is a more durable toxin than microcystin. So I think that's why we tend to see it in those clear lakes and then in the cold water temperatures.
Thank you for that. And I see that Val just clarified her comments, so sorry about that, Val. I am not the subject matter expertise, so I apologize if I misinterpreted that question. Well, we are right about at time, so I'm gonna go ahead and share my screen here and just give you once again both of our presenters' emails. If you did have a burning question that you weren't able to get answered today, I wanted to provide that. Should you want to reach out, that also is a great avenue to look them up and see about their labs and a little bit more of their research and work. Today's session, as a reminder, was recorded, and we will post that to our website at northcentralwater.org/HAB. So be sure to look for that. Seeing lots of kudos come in for the presenters. So thank you both for joining us today and for your time and talent. I do want to let folks know, actually next week we have another webinar. This is part of the current webinar series, and we're going to be highlighting a couple multi-state water projects that have been really successful. It's actually the 100th edition of the current webinar series. So if you're available, please join us for that. We'll be talking about conservation drainage and watershed leadership, as well as the upcoming North Central Agriculture and Climate Conference. So thank you all for joining us today. Thanks to our speakers for lending their time and expertise, and we hope you can join us at a future edition of the Algal Bloom Action Teams webinar series.